And so we're going to shift from I think one of the key goals for our community is that I think many of the insights we had initially were clear. We knew that this was an important thing to do, and as a community we came together and said, we really want to do it, but there's a difference between the why and the how. And so after we know that we want to do something, there's still an open scientific question as to how to do it, and there's also an organizational question as to how to do it. And in both of these cases, we took the approach of taking this year-long planning process, traveling the world, convening the community in many different places, and keeping the door always open to anyone who actually wanted to participate, and also engaging the community in actually generating, in the end, the blueprint or the plan of how to build an atlas. And as all scientific plans are, there's a plan in the beginning, and then you actually do things, and the plan changes. So the first thing to remember about the white paper is that it is a living document. It's never going to be published in a journal. That's not its goal. Its goal is to live online and be periodically updated. There are things that are blank there and are left blank purposefully. It says, here we will have something about the model organisms, because the model organism community is only going to meet in maybe 3 weeks or so in Berlin. And they're going to discuss, and then they're going to write a section, and then the community is kind of going to iteratively think through it until it feels like the right plan, and we move on. So we try to summarize the how-to in one figure. It's not one that one can parse out in a single slide, and I'm actually going to take you through this stepwise over a series of slides, as we start from a human and end with an atlas. And the first thing that we start with is our specimen, or where do the tissues come from, and what is the atlas going to cover. And so the first decision we actually had to make as a community is at which level of the body is our first draft going to happen. And everything that I'm going to describe is going to be about the first draft, not about the finished atlas. But we wanted to have a milestone that was reasonable and achievable, and at the same time had a lot of information in it, as Sarah conveyed to you a couple of slides ago. And so one could think about doing every organ and every system, and there are two questions here. Which organs and systems might one go after first? And at the same time, within an organ and a system, at what level of resolution are you actually going to capture it? And one of the decisions that we made was that we are not going to reconstruct a whole organ, so also understand the organization of the full organ, say the whole brain or the whole gastrointestinal system, but instead we're going to work at the histological level, meaning we will sample the different parts of the anatomy of an organ, and we will know what they're composed of, but we might not know how they all come together, for example, and finally make the full brain. That would be in the Human Cell Atlas, but only in the second or the third draft, not in the first one. Then we had to ask ourselves, which organs and systems? And when you think this question, you have to take into account different considerations. So one of them is that you don't want to be too limited. And especially you don't want to be too limited to the easy part. You want to kind of cover things of different natures and identities, so you get a good sampling of the whole problem. So doing the brain is not the same as doing an epithelial tissue, is not the same as doing a distributed system like the immune system or the skin. So you have to think about each of these kinds of things, but you might not take many, many examples of each of them. And so after a process in the community that involved also people basically standing up and advocating for their system, we came up with 12 organs and systems: the nervous system, the immune system, the kidney, the lung, and the trachea, the liver, the pancreas, the small intestine, colon, and esophagus as parts of the GI, the skin, the heart, and also these 3 areas of developmental biology, which is the developing human embryo and fetus, pediatrics, not as a full scope pediatric atlas, because that would basically be a replication of the adult human cell atlas, but at least starting to look at pediatric tissue early on so that we do not forget our children behind. And organoids is an area of regenerative medicine where one would want to compare a system that is taken outside the body to the initial tissue. And so in each of these areas formed biological networks, and they really formed themselves. We might have gone occasionally after an individual or two and dragged them in, but most of the time people just said, I'm really interested in the heart. Is it okay to plan for it? And we would say, of course, yes. And so these groups of individuals actually came together, scientists from all across the world, usually working across institutions, across countries, and came up with their motivations, their key considerations, what might a pilot look like, and what would be the plan for the first atlas. And so these are all laid out in the white paper in individual sections. Then there's the question of where do the tissues come from? And it is interesting to understand that throughout this entire scheme, I'm going to come back to this notion of the trade-off. One thing that became very apparent through the repeated conversations in all of those meetings is that pretty much at every different axis, we have a trade-off between two different things. And so we have to find ways of balancing that trade-off. For tissues, the trade-off between those tissues that you can obtain fresh from actually healthy living humans to the ability to comprehensively cover the whole body. The brain is a great example. You are not going to sample healthy brains from living individuals. You might get a bit here and there, but not in any comprehensive way. Whereas on the other hand, you could obtain them, the full brain, in a post-mortem examination. Other things you might be able to obtain from transplant donors, not the brain, but other tissues. And they are closer to healthy, and they are fresh tissues. And finally, some tissues you can sample at the level of a biopsy or a resection because of other clinical needs that arise. And so there's a spectrum there, and we have to accommodate that spectrum. And that means that in the full scale, we have to collect in post-mortem, because there are things that we would want to collect from a single individual, every single tissue, and we would want to collect whole organs so that we would have appropriate sampling. But in certain instances, we can go one step back and go to transplant donors. And in certain instances, we can even go to truly healthy individuals in our community. And so those cases where we can go all the way through will give us a sort of a Rosetta Stone to tell us whether the postmortem is actually appropriately representative or whether there are actually issues there that we need to grapple with and solve technologically in order to obtain appropriate information about the human body. The next layer for tissue acquisition is how many individuals should you sample? I think this is the question that gets asked the most, right? Yeah. Sarah is nodding. This is the question that gets asked the most. And the reason for this is that we're used to think about cohort studies in humans as something that requires a very large number of individuals, and we usually call this N. N will be our number of individuals. For a study such as the Human Cell Atlas, when we're trying to build a reference, we do not expect to cover that number of individuals. And this is very similar to thinking about the Human Genome Project, where first the Human Genome Reference was built, and in parallel, and definitely forward-looking after that, the human genetic diversity was really grappled with in its full scope. This does not mean that we're not committed to diversity and equity and inclusiveness, as Sarah laid out. These individuals that will be sampled will be diverse, but we will also expect at the same time that their number will be relatively small. And so most of the communities have converged on the number of maybe between 20 and 60, based on admittedly a limited amount of information that we have in our hands today. So for example, we have seen both in fresh, healthy tissues, I'm showing here colon biopsies, and on the other end, in brain from postmortem, that looking at about 10 individuals actually gives highly reproducible results across them. And these were not individuals that were particularly selected. So going a little bit higher and across multiple sites might cover for us sufficiently what we would call a reference without an extensive understanding of the role of genetic diversity and also experiential diversity at impacting our cells. That would be for future studies after us. We are focusing mostly on adults, although we saved room for both human development in a pediatric atlas, in a fetal cell atlas, and for pediatrics in a pediatric atlas. We know that the large number of cells might help compensate for the smaller number of individuals. So far, inter-individual reproducibility does not seem to be a remarkable issue. And one of the technologies that is, or one of the experimental strategies that has really been pioneered in the community recently has been the pooling of individuals early on, which allows you to actually analyze maybe more of them than we initially thought. And that's an example of how we will have to be technically and intellectually flexible. An idea like this would come up, we might revise these numbers, especially for those cases when we can actually afford pooling. And we will get to the same number of cells, but immediately looking at more individuals. And there are members of our community, for example, John Marioni and Jimmy Yee and Oli Stegley, who have actually pioneered this idea over the span of the exact same year in which we were, planning an atlas. The third aspect in thinking about our samples is the fact that humans are different from each other. I'm not particularly tall. Stan Linarsson is. Our organs are not scaled in exactly the same way. Sorry, Stan, for putting you on the spot. You will see, he'll stand up here. I am way shorter. And so our organs are not scaled in the same way. And as a result, taking a specimen from one individual and saying that it is the same, that this is the same thing in some abstracted notion is difficult. This is something that in the field is called the common coordinate framework, into which you want to harness everything back and say that it's the same. There is a lot, and again, there's a trade-off. On one end of the trade-off is prior knowledge. Anatomists have been working for many, many centuries, right, at mapping the human body and abstracting it in this way and writing the textbooks and so on. So we can use that prior knowledge. But at the same time, we know that this is actually difficult, that at the gross anatomical level, it's easy. At the fine histological level, it's quite easy. And somewhere in between, it gets really challenging. And so there's an opportunity also on the other end of the trade-off to be very data-driven about it. And if it's data-driven, that it means that in advance, when we actually collect the specimen, we can't say where it came from in this abstracted notion because we don't know. We're going to discover it. And so what becomes really important as a result is something very operational, to be able to track the samples as we go in a very physical sense, to know that this little bit of tissue that a lab, for example, received for analysis came from this very exact, precise place in a particular specimen. So that was a lesson that we learned this year from discussing that. Then, once the samples arrive, we're going to go back to our image and think about what are we actually going to do with them. And so we define two major branches for the analysis in the atlas, and we call them the cellular branch, sometimes also called the nuclear branch, and the spatial branch. And these are different styles of approaches that provide us different information. One of them gives us information on individual cells or their nuclei, so dissociated from their tissue context, and the other tells us about how they're organized relative to each other in space. What is absolutely critical to know is that these are two equal branches for the atlas. Sometimes people are deceived by the name, the human cell atlas. The identity of the cell relies also on their neighbors and on where they are positioned, just as much as it relies on what is intrinsically happening inside it in terms of its molecules. So they are on an equal footing. And again, I think it is an example of our community reaching this realization that this is actually a practical thing to do. A year ago, when we met in London, a lot of the techniques that would be needed for the spatial branch were kind of emerging all at the same time, and people were not sure, would they be ready, would they not be ready, should we do it, are cells enough? And at some point, by the middle of the year, it was absolutely clear that everyone wants both, and that was great. That was an easy solution in a sense, but there is a hitch. There is a difference in the step of these different technologies. So the cellular branch methods have, I would say, not just matured, but especially disseminated in the community very dramatically in the last couple of years so that everyone can use them. And that means that it's easy to go with them right away. Whereas the spatial methods have had a renaissance of technological innovation, which is actually quite mind-blowing, but they're still not as widely disseminated, and some kinks are still being ironed out. But they have a huge edge. Their huge edge is that you can apply them to tissue that has been preserved, either frozen or fixed, depending on the technique. And that means that we might not necessarily have to decide tomorrow morning on which technique we would use. We could wait a year. During that year, methods will stabilize. There are a lot of, for example, projects that have started now to compare different methods and optimize them and so on. And so we could come back to these specimens. And again, that emphasizes that we will analyze every specimen by both approaches, but we might actually do this analysis in some temporally staggered way. Why do we want to have both? One of the reasons that we want to have both is the fact that they each report on something different. It also turns out that they're mutually beneficial to each other. And so spatial transcriptomics, information, for example, about where transcripts are expressed in space, but not necessarily knowing exactly in which cell they are, can become single-cell resolution with additional single-cell data from the same system. And, excuse me, everyone was here in SCG knows that I, unfortunately, have a cough right now. And so spatial targeted assays require us to know a signature of genes to assay in the tissue, and the single-cell profiles can help us know what these genes are. But once we do the measurement in the tissue, we can actually take the rest of the profile and map it back to the tissue. And so there's nice mutual benefit between the two. So I'm going to spend a few minutes on each of these branches. I'll start with the cellular branch. The idea for the cellular branch, and this is a term that we stole shamelessly from Christophe Benoit, who suggested it, I believe, in August, actually before the London meeting. He calls it the skydive. He says, if you want to understand a very complex population of cells, think about yourself as going through a skydive. You start at the top of the skydive. You're looking very, very broadly. You see everything. But it's, not the best exact resolution, right? Because you're not close enough to it. And as you understand what the landscape is in general, you might decide, okay, these are the 5 pieces of it, and I want to zero in on each of them. And so that is what we will call the stratified approach. And, he's an immunologist. Immunologists like cell surface markers, and they sort. But there are different ways in which you can zero in. And so you zero in into subsets of cells predefined by their molecular attributes. And once you do that, you can go through this iteratively, because the more you measure, the more you know about the entity that you're actually measuring. And so this maps up to this kind of 3-phase process where we start in a uniform way using the most massively parallel approaches that we have. That handles, again, a trade-off that we have between the number of cells and the molecular depth that we have in each of these cells. We can do a lot of cells quite shallowly, or a few cells but very deeply. And as we walk down in a more and more informed way, we can invest more and more into the cells that we're looking at. And so in order to know how to transition through this, we actually need computational analysis, and in particular we need statistics. We need a power calculation that will tell us what is the number of cells that are needed in order to detect all the type at a given rarity. And again, I'm going to allude to what has happened in human genetics. In human genetics, people have not said, I'm going to find all the variants in the population that matter. They said, I'm going to find all the variants up to, say, 5% or down to 5%, and that's what I'm guaranteed to get. And that's because they had power calculations. And the same is true for cancer genetics. And we can take the same set of ideas. Of course, the power calculation has to actually be completely different, and use that, and one can introduce additional criteria like cost-benefit analysis. In the spatial branch, again, we have a trade-off. We can have high resolution today with some methods, or we can have genomic scale with other methods, but we currently, at least, have not found yet a magic method that gives us both. We might, in which case the trade-off will no longer exist. But today, we kind of have a balance between these two. And so the best approach is to actually do both at the moment, because each gives something distinct. And as I pointed out, because this is a rapidly evolving technology landscape, it is critically to bank the specimens. And then finally, while we are most definitely focused on the high-resolution data, we do realize that there is auxiliary data that one may collect that would not be necessarily in the core of the atlas, but would be very important for its interpretation. So one can imagine EM images of cells. That has been very effective for neuroscience. Histological information will be important. Bulk RNA-seq data helps us annotate the transcriptome very effectively, against which we peg later on all of our quantifications and so on. There are many, many things that can be in this list. It's also our commitment and opportunity to engage with other consortia and initiatives like ENCODE or the 4D Nucleome, the Human Protein Atlas, with whom we can share in this particular mission. So we can share specimens, we can share reagents, and we can share approaches and data. We need to know that whatever we collect is actually correct, and so validation is an important point. For people in the field, they do know that when, say you found a cluster of cells, in some sense, so what? How do I know it's a thing or what the thing is? And so we considered this, and it's a challenging problem, and we considered three approaches for validation, or three criteria. The first is that whatever we recovered is reproducible. That's the minimal bar to pass. So you want to recover it again, say in two independent sites, and you might also want to have the ability to do prospective isolation. If you identify some molecule, for example, expressed in your cell, if you only isolated cells with that molecule, will it give you one-to-one matching to the initial place from which you started? So multi-site and multi-individual replication studies are going to be important. The second is what we call the integrity, especially for single-cell methods. We do know that we might encounter loss in the tissue, meaning as we process the sample, we might lose, for example, some of the cells. And if we lose them, we might not even know they were there. How would we know that we actually captured everything? And so one of the best ways of addressing this is comparing it to bulk signal and seeing whether there's residual signal in the bulks that we can discover. The other major approach to distinguish this is by comparing across modalities. For example, using single cells and then going and staining a tissue of the same kind and seeing whether there's something I see there that is not covered by anything here. And then finally, we can ask about predictive value. I think we have a limited capacity there, but there is some. We can determine a subset that is distinguished by one set of features, for example, gene expression, and see when can it also fit another set of features, for example, morphology. That sometimes works and sometimes doesn't. And again, the cross-referencing between these two branches is absolutely critical in order to get this notion of predictive value. I would say that for a lot of biologists, predictive value would involve actual functional assessment, for example, perturbing a system and seeing what happens. That is beyond the scope of the atlas, most definitely beyond the scope of the first draft. And again, by analogy to the Human Genome Project, people have discovered genes in the Human Genome Project. They didn't immediately know what all of them do. And in fact, even now, we don't know what all of them do. And so that was an appropriate choice, because had they been bogged down with characterizing the function of every gene as it was being discovered, we still wouldn't have had a human genome today. And so we drive the atlas in two technical ways. One is experimental technologies, and the other is computational technologies. We bring the groups. So first, starting with the technologies, it's important to understand that technologies have a life cycle or a lifespan. First comes the need. Then people come up with an invention. They go through a process of tech dev. They provide a proof of concept. Often after the early proof of concept, there is a need to optimize and to scale the method. And then you want to get it to really work in kind of a scaled way. And you also want it to be very disseminated. There is a difference between doing a lot of it yourself and having everyone else in the world use it as well. And I think for our field, what has been transformative is how quickly everything disseminated to the world. And so the goal of what we call the technology forum is to really bring together the groups and the centers to compare and disseminate existing methods, and also to push people to ensure that they develop the new methods and adopt them. For example, inspiring them with new needs and saying, that's really important for us. If you can come up with a great solution for this, it would be very transformative. And then establishing the benchmarking standards and sharing reagents and having Rosetta Stone samples that you can come back to again and again and use with the next method and see how it compares to earlier methods. And that applies to both the cellular and the spatial branch. Again, like the cellular and the spatial, so are the technical and the computational. Computation starts in the human cell atlas before a single cell or tissue have been analyzed at experimental design. And so there are multiple areas in which we have to have substantial computational innovation. One of them is the core questions. What is a cell type? How do we understand the spatial dimension and how cells are organized in their tissue? How do we understand the temporal dimension and how cells change over time? And the flip side of that is the very practical side. We need to process and normalize data. We need to segment images. We need to design experiments, and we need to do it hand in hand, and we need the right power analysis to drive these iterative schemes. And we need to serve data to the world, not just in the sense of having it well organized, we'll get to that in one slide, but in the sense of having great portals and great ways for biologists to ask their question of the atlas and to get benefit out of it. One can think of the core of the atlas as this massive data set, and a massive data set deserves its own data platform. In the Human Cell Atlas, this is called the Data Coordination Platform, or the DCP. And as Sarah said, that was pretty much the first thing that we started thinking about, even before knowing what would be the overall contours, because it was clear that if, when stuff starts arising, there isn't somewhere to put it, we're all going to be in deep, deep trouble. It was also clear that that thing does not exist, and that was actually a huge benefit for our field. We have no legacy, so we're not stuck with anything. We can design from scratch. And so a team of groups came together, and this involves groups from the EBI Sanger, from UCSC, from the Broad, and from the Chan Zuckerberg Initiative, and Jeremy Freeman is up there, he can wave. He has a cool laptop. And in an iterative process, came up with a scheme that really tries to build on the latest and greatest in software engineering architecture and the unique nature of this data. And thinking through this scheme, try to come up with a design that would be particularly modular and flexible, but extraordinarily robust. And so, first of all, we had a set of principles. We said that we would want to have a single data platform for HCA, not some federation, a place. We wanted it to be open source and fully clonable, meaning somebody could come, grab the whole thing, either the whole code or the code and the data, and just dump it somewhere in their own world and do whatever they want with it. We wanted the data to be open access, but we wanted to assure that there would be infrastructure for what we call controlled access. For example, if a disease area would want to come in and would have restrictions that are required by ethics, and that's our commitment to ethics and privacy. We wanted it to have open APIs so that anyone can build a portal for either analysis tasks or visualization. Anyone in the world, that's what we call our analysis garden. We wanted it to be developed entirely in the open, and it is. There's a URL there on GitHub. If you want to, you can go. As I noted, the developers currently are from UCSC, EBI, CZI, and the Broad. And there has been very rapid progress because I actually was in Palo Alto 2 weeks ago and saw a live end-to-end demo where people from these different places who are placed in very different geographical locations actually ingested data all the way down to processing it and visualizing it. And that was kind of cool, since they started in June, by the way, and there was nothing before. And so let me tell you a little bit about how it's organized. It has these very colorful parts, and that's how they're called. They're called the purple, the blue, the green, and the red boxes. And so I'm initiating you into the color code. The purple box is the ingest. This is where users actually come and upload their data. It is responsible also for all the metadata, all the information beyond the data itself that describes the specimen, and this is being developed by EBI. Separately from that is the data store, which resides on the cloud, currently on 2 commercial clouds, and eventually will be on 3 commercial clouds. So Amazon Web Services, Google, and Microsoft Azure, and as I pointed out, can be cloned, and is designed purposefully not to be married to a particular vendor and not to be private. The data store stores all of the data. It stores all of the raw data and metadata, but it also can store certain types of process data. This brings us to the green box. There is a certain set of what we call HCA approved or blessed pipelines, which are for certain types of analysis tasks that we consider to be both routine and where standardization is particularly important. And so data that runs through these analysis pipelines feeds back to the data store and is available in processed form. You can think, for example, if you're thinking single-cell RNA-seq, reads get mapped and expression gets quantified. If you think on an image, cells need to be segmented. But that's basically it. That gets stored back in the blue box, in the data store. Everything else people want to do, find your favorite trajectory, for example, in data, will happen in these red boxes. These are all called portals. They do both visualization and querying tasks, but they also do a lot of analysis tasks, and they're built by anyone who wants to in the community. No one gets anything special in building a portal, including the DCP developers themselves. And so for the last couple of minutes, we're pretty much on time. So for the last couple of minutes, I'm going to talk to you a little bit about the other piece that you have to think about when you're writing a blueprint for an atlas. One part of it is, what would we do as scientists? We've just covered that. We have a plan. It will change. We know it will change. But it's a plan we can get started on. It actually tells us, do this, do that. And who said that? We ourselves. But at the same time, we also need to have organization. And this is an unusual organization, the Human Cell Atlas. It's very open. It wants to keep the doors open. It doesn't want to introduce too many restrictions. Most of the restrictions have to be about how you have to be open. But it wants to learn also from past experiences and then apply those to what is the challenge that is at hand. So first we kind of turn to what has been successful in the past. I'm not going to belabor the whole bulleted point. And then based on all of these lessons, we went and started thinking about our organization. So I'm going to take you again through this org chart, as you might want to call it, through the different components of the consortium, step by step. I'll parse them out as we go along, but I'll call out the main issues up front. We have an organizing committee that actually Sarah and I co-chair, along with 25 of our best friends, so altogether 27. I'm joking. There is the organizing committee has executive offices that help run its standard operations. It has working groups. It has a governance group for the data platform, and it has a registry. And we're going to go through each of these components one by one. So I'm going to start with the organizing committee, also called the OC. It's our decision-making body. We actually have to make a lot of decisions, both scientific and organizational decisions, and it is this group that makes those decisions. It's fully populated. We envision about 35 scientists, and it could expand for geographical representation, for needed expertise, to ensure gender balance and general diversity. The new members are added by majority vote. Members have 5-year terms. It has 2 co-chairs. Both of them are members of the OC, and they also have 5-year terms. It has an executive committee. It is made of the 2 co-chairs and 5 additional OC members, and they have 2-year terms. And they're responsible for a lot of routine tasks that one needs to do quickly between OC meetings, and they prepare the meeting agenda, and they provide guidance. All the terms can be renewed once by majority vote of the OC, and we would periodically seek input from the HCA community on the member's scientific scope and performance. What does the organizing committee actually do? It has to convene the community. For example, this. We have to invite people to meetings and to workshops and to jamborees, and we do that. We coordinate and we author key documents. For example, we coordinated the writing of the Human Cell Atlas White Paper. We define scientific values and ethical principles. We make sure that we both define and uphold the necessary processes, including QC standards and analytical standards. We're responsible to governing the data coordination platform. I'll talk more about that, and the Common Coordinate Framework. We define the criteria for membership. We coordinate the work products. We communicate on behalf of the HCA. We represent and negotiate. and we poll the community on all the issues, including our own performance. I'll give you a few examples of things we actually do through two different groups that are established by the organizing committee. One is the DCP Governance Group, so the governance group for the data coordination platform, also known as the DCP-GG. The organizing committee has to make policy decisions for the DCP, like what would actually be the pipelines. And it delegates those downstream, and it makes sure that once the policy decision is made by the organizing committee, others follow through. And so we approve an overall plan, and then we appointed a group, the DCP-GG, in order to oversee that the policy gets appropriately implemented. The chairs of the DCP-GG are Roland Ailes from Germany and Udi Shapiro, actually here from the Weizmann. And the key topics that they have to oversee are the definition of the data manifest, meaning what's the actual human cell atlas, official analysis pipelines, the required metadata to reflect data collection standards, the common coordinate framework, and any formal release portal. They are responsible to seeing that any decisions that the OC or a working group made with respect to these items actually get through to the DCP. We also have working groups. The organizing committee establishes them, and they mandate them to take on specific key areas. Each working group, again, has two co-chairs. We found that these two co-chair thing really works for us, and so we have continued with this. One of the HCA working group co-chairs is always an OC member, and the other is always not, so it's external. The additional members are selected by the co-chairs. And currently, amongst the groups, there's an analysis working group. I have a small typo there. A metadata working group, a working group for the common coordinate framework, a standard and technology working group, and an ethics working group. I'll give you an example. The first working group we established, similar to starting thinking about the DCP early, we also started thinking about analysis early, and this was the first one to be established. Dana Peer and John Marioni lead it, Dana from the SKI and John Marioni from the EBI, and its members are Peter Karchenko, Rahul Satija, Oli Stegli, Amos Tanai, Fabian Thais, and Nir Yosef. And they're responsible to bring the community together. They conduct jamborees and workshops. They are responsible to deciding that this is a good pipeline to actually introduce for read mapping, for example, so to bless the standards and the pipelines and the analytics for the DCP. And they do this through transparent and systematic evaluation, not just by having a conference call and deciding that they'll do it, but actually by engaging the whole community in this process. They are engaged in experimental design and in new analysis methods for the tissue atlases, and they are responsible to conduct community-wide activities to engage the community and also to disseminate and educate it. So you might be very engaging within the computational teams, but if you're not also disseminating broadly and educating the overall HCA community, then you are not as successful as a computational scientist. They've held a remarkable first jamboree in EBI in August. There were 6 challenges addressing many of the vexing problems that people actually struggle with in our field. They had 48 young scientists from 19 computational biology labs. These were assigned to groups. No 2 members of the same lab were allowed to be in the same group together, so they had to be in separate groups from each other and work from people from other groups, in which case you're making the community work better together. They ended up with 5 GitHub repos with over 100 commits. And in fact, they've made a substantial improvement in algorithms for different challenges, for example, for doublet detections. And they're busy writing papers now with better methods than any of us came up with alone. And that's kind of cool. And then we can move on and work on other problems. Who's in the HCA? At first, this was not a well-described notion. And so if we're after describing ourselves, we'd better be able to say who we are. And so we have now a formal process, both for membership and for projects. Any individual may become an HCA member as long as they register. At registration, they have to agree to abide by the principles of the HCA, the values that Sarah described to you, and especially the ethical standards. And they get invited to attend HCA community meetings and discussions and basically engage in the activities that we have done so far. They can join the HCA Slack channel. They will be on mailing lists. They will get an insight into what is going on live. In addition, there is a second tier, which is what we call an HCA collaborating member. And these are those members that have already engaged in actual work in the HCA. And this could be either because they are a participant in an HCA project, or because they are a member of one of the groups that the OC designated. In fact, some of our members are such that they might be giving to the community more than they would actually be taking out, and we wanted to make sure that this generous contribution is appropriately designated. We also have HCA Projects. Any scientific project that is related to systematic biological characterization at single-cell resolution that involves cells and their spatial organization may become an HCA project. This includes data generation, but not just data generation. You can be an experimental method developer or a project to develop a computational method in this domain, and that would be an HCA project. There are requirements. You have to register your project, where you have to describe your project and its strategy and its investigators. You might want to describe you might want to describe additional information about it, and you're also to commit to give something in return. The minimum is that you're committed to the HCA standards and the information release policy. I'll explain that in a second, but they come in 3 categories. A participating project is basically someone registering themselves and saying, I'm doing this, and I'm committed to your principles. You say something about the content, the participants, and the funding, and you're done. A network project is also committed to interact with others. They don't have to agree on what they do. They don't have to be connected to do something together, but they have to communicate with each other and have real scientific, meaningful interactions with each other. And then a flagship project is a network project that aims to deliver a whole component of the draft atlas. For example, the immune cell atlas is a flagship project, and that involves substantial funding. There is a registry where a person can actually go online, and I believe that it is live now. So join HCA. We will, of course, remind all people who have attended any meeting and that we consider members of the HCA to register. But this would also make it simple for people who ask that question, but how, to have a tangible way of coming in. 2 last slides. Our information release policy. We said multiple times open access, but we're truly committed to this. And so all HCA projects commit that their data is deposited in the DCP through a regular data release. The data is available in an open access manner to the maximal extent that's allowed by ethics. The DCP will tag the data, so it will become citable, for example, with a DOI. The users will be free to use the data as they wish. No restrictions, no limitations, no I have to publish before you publish. Everyone will be able to use it the moment that it is out there. They only have to cite the generators of the data, and they have to be committed not to try to identify individual research participants, which is an ethical requirement. The organizing committee will work with journals to define opportunities for key data collection and analysis papers, and we are committed to publicly releasing also all the experimental and computational methods and source code used. And so far, we've had a great interaction with the funders around this. For example, in the CZI projects that are coming now online, this has been a requirement from all of the grantees. We don't just say this, we practice what we preach. And so in a couple of weeks, the first little snippet of an HCA project will be out completely open access immediately after QC has been finished and it's been uploaded online without anyone actually having had a chance to really deeply analyze their own data. This will be about 1 million cells. All of them are immune cells. They will come from relatively simple sources of immune tissue, such as cord blood or bone marrow. You're looking here at the actual cells. This part is the, I believe, peripheral blood, and that part is bone marrow. You can see the different individuals colored there, different cell types colored there, and it looks like an unbelievably gorgeous picture. Now, you might have noticed that we gave no credit to no one on any slide, but I felt compelled to thank these individuals because these are basically postdocs and scientists who normally would want to write a paper about their cells. And instead of writing their paper, they're putting their data out. They'll also write papers, but they're putting their data out. And I think for the organizing committee and for the members of the HCA, this will become second nature. And it will change the nature of our field, and it would be very exciting. And in this, we hope to achieve, even broader participation and reach than we have now. We're pretty good. We're on all continents. We're still working on Latin America, but we have everything, including Africa. And we're quite excited about this participation. And with this, we will finish, and we will invite Stan to lead the discussion. Thank you.